Hello, my name is Toni Hauptmann. I am a researcher in the data mining group at Johannes Gutenberg University Mainz, and I'm going to present you the results of our comparison of multi-omics integration architectures. The research on multi-omics integration with deep learning started a couple of years ago. The literature about it is growing steadily. However, close to nothing is known about relative performances of integration methods. The reason is that many authors don't compare their methods with others, and if they perform a comparison, it is only against a weak baseline, or they favor their method in the optimization. Our research objective was to compare deep learning architectures for multi-omics integration under fair conditions and determine the preferred method for different use cases. For this, we performed experiments with a comparison framework we developed. The experiments revealed that statistical significances between the methods hardly exist, despite trends in their rankings. Our framework is relatively straightforward. We performed the 5x5 nested cross-validation, whereas our methods use the same hyperparameter grids for shared variables and individual ones for exclusive hyperparameter. In the inner cross-validation, we performed 200 iterations of random search to optimize the hyperparameter. We used AUROC and area under precision recall curve to validate different aspects of the architectures. In our experiments, we used 7 publicly available drug response datasets. Each of the datasets contains cell line samples and additionally either PDX or patient data. For each sample, 3 different omics are available. These are gene expression, somatic point mutation, and copy number alteration. The cell line samples were used in the nested cross-validation to train and optimize the models. PDX or patient datasets, which contain too few samples to be used for training a neural network, were used as external test set. Here's an overview of the 7 multi-omics integration architectures we compared. They can be broadly categorized in 3 categories depending on the integration depth. The first category is early integration, which concatenates omics to use existing methods for single omics analysis and which assumes a high dependency between omics. The second category is intermediate integration, which first encodes the input samples to a lower dimensional representation before concatenating them. This has the advantage that it does not increase the dimension of the input space. The last category is late integration. Late integration architectures analyzes omics individually before combining the individual results into the final one. The combined results can be simple as an average or the result of another machine learning method. Late integration uses the same methods as early integration, but assumes less dependencies between omics. During the work on the comparison framework, we also developed an architecture that combines intermediate and late integration. Now I'm going to zoom in on individual architectures and explain them in more detail. The first architecture is early integration. As mentioned earlier, it has the advantage that it's simply to use, but it assumes high dependencies between omics. One concatenates all feature vectors, which makes it possible to use existing methods for single omics analysis. But in the context of multi-omics integration, where currently only small datasets exist, early integration has the disadvantage that it increases the dimensionality of the already sparse input space and therefore making it even sparser. In our experiments, we implemented the encoder and classifier with fully connected layers. The next architectures belong to the category intermediate integration. Whereas early integration assumes a high dependency between views and late integration a low dependency, intermediate integration represents a trade-off between them both. In intermediate integration, the views are first transformed to lower dimensional representation with a suitable encoder. Only then, the transformed representations are concatenated and evaluated. The first method I want to present uses principal component analysis for encoding and a neural network as classifier. The omics are first encoded individually with PCA to reduce their dimensionality before they are concatenated. Dimensionality reduction with PCA is unsupervised and linear and could not be able to capture nonlinear dependencies between input features. Therefore, the following methods use neural networks for encoding and classifying. The first of these methods is MOLI, short for Multi-Omics Late Integration, which is, despite its name, an intermediate integration method. The novelty of MOLI was that it additionally uses the triplet loss for training, which was originally developed for face recognition and which is well suited for biased datasets that contain underrepresented classes. The idea of triplet loss is that encoded samples with the same class should be gathered in the same subspace and that the subspaces of different classes should be pushed away from each other. As the name implies, one needs to first create sample triplets. Triplets are created by randomly choosing two samples of one class and one sample of the opposite class. One sample of the twice strong class acts as anchor element. The remaining sample of the same class is called positive element and the other one is called negative element. After creating the triplets, the distances between the anchor and the positive and negative element are calculated. The distances difference is used to determine the loss value. If the difference is greater than the chosen hyperparameter alpha, the difference to alpha is the loss value. Otherwise, if the negative sample is already far away enough, the loss value is set to 0. Supervised feature extraction learning using triplet loss, called Superfeld, is based on MOLI, but instead of training the encoders and the classifier end-to-end, the training is split into two distinct phases. The first phase trains the supervised encoder with triplet loss for each omics to extract latent features of the high-dimensional omics data to avoid overfitting. After that, in the second phase, the encoders are fixed and used to encode the input samples. The encoded omics are concatenated and used to train the classifier. The author's idea is that disentangling the loss functions puts the focus on only one task at once, which shall increase the prediction quality. The next architecture is OmiEmbed, which is again an intermediate integration architecture. Its neural network is a supervised variational autoencoder. The method learns the latent features with the additional unsupervised task of reconstructing the input omics. The trained latent features can be used for one or more supervised tasks. The autoencoder is trained in three distinct phases. In the first phase, only the encoders and decoders are trained without a classifier. In the second phase, the variational autoencoder is fixed and only the classifier is trained. In the third and last phase, a low learning rate is used to fine-tune the autoencoder and classifier together. Multitask Attention Learning Algorithm for Multi-Omics Data, MOMA, is a late integration method combined with an attention mechanism. The visualization for 3 omics would be too complex, therefore I only show the architecture for 2 omics. MOMA uses 2 neural networks to encode each omics in 2 different ways. Both feature vectors are stacked and interpreted as a collection of 2-dimensional vectors, which the authors called modules. The module attention component creates weight matrices, which are used to generate the final modules. The weight matrix entries are defined as the cosine similarities between the models of 2 omics. All similarities of one module are transformed into a probability distribution with the softmax function. 2 weight matrices are generated for every combination because the generation is non-symmetric. The final modules are defined as the multiplication of the modules with the weight matrix. The flattened modules are used to predict individually the drug response. The predictions are combined with a logistic regression to get the final prediction. The last method want to present is omics stacking, which was developed by us. We wanted to combine the advantages of intermediate and late integration. Omics stacking preserves the weak signals in omics, which could vanish during the omics integration by training individual classifier. But to also capture and model dependencies between omics, another classifier is trained on the concatenated latent representations. A meta-learner, which is implemented as a neural network, learns on its own the best way to combine the results into the final prediction. It could, for example, learn which classifier gives precise results and can increase the weight for its results, or ignore omics that does not provide information for the current task. Like Molly and Superfeld, we are using tripled loss to balance the biased classes. After explaining briefly our architectures, I will now present the comparisons results. This table shows exemplary the mean ROC for the cross-validation test sets. To specify the best method per use case, we first performed the Friedman test to verify if the mean of at least one method is different. When a difference existed, we additionally performed the Nemini post-hoc test on the method's mean ranks to define the critical difference. The results of the post-hoc tests can be visually presented with a simple diagram. The top line in the diagram is the axis on which we plot the average rank of methods. When comparing all the algorithms against each other, we connect the groups of algorithms that are not significantly different. We also show the critical difference above the graph as reference. The performance of two classifiers is significantly different if the corresponding average ranks differ by at least the critical difference. Here are shown the results for the cross-validation. The only thing we can say is that Superfeld is significantly better than PCA. But for the other methods, we do not have enough information. Another conclusion is that PCA and early integration tend to achieve worse results. The results for the external test set are similar regarding significant differences. But in this case, omics stacking is significantly better than early integration for the ROC. For area under precision recall curve, the Friedman test already couldn't reject the hypothesis that all means are the same. Hence, we didn't compute the critical difference. But one noticeable trend is that omics stacking achieved the lowest rankings for ROC and area under precision recall curve. Again, early integration is belonging to the highest ranked methods. Now I'm coming to our conclusions. It is important that researchers who develop a new method perform a comprehensively comparison to contextualize their method's performance. More and larger datasets are needed to reduce the variance and to increase the power of statistical tests. But the problem is how to share omics without violating data protection. If that task is solved, a lot more datasets which already exist locally could be shared. Our experiments were limited to drug response prediction, but the results may vary for other tasks. To ensure comprehensive future comparisons, it is essential to incorporate a diverse range of tasks. When researchers are faced with a new dataset, we advise them to consider early integration or PCA solely as baseline methods. This is because its approaches will, particularly for small datasets, perform worse than an intermediate integration method. Additionally, we suggest employing Superfeld for cross-validation experiments, where no predictions on an external dataset are made. However, if the objective is to generate predictions on an external dataset, we recommend utilizing omics stacking instead. If you are looking for further information, check out our paper and the source code.